Our next speaker is Tony Purcell, professor at Monash University. And with Tony's presentation, we're going to learn how utilizing a research-grade Zeno-trap time-of-flight instrument can advance applications in immunopeptidomics and proteomics, including giving us an overview of what ZT-scan DIA is, preliminary proteomics and immunopeptidomic results using ZT-scan DIA, and the future applications. So with that, I'll turn it over to you, Tony. All yours.
Thanks, Jamie. We've had a few technical issues here, so hopefully you can all see my slides, and we'll get through this unscathed. So I just have a few disclaimers, and I guess the only really relevant one is the collaboration that I'll discuss today with SCIEX. So as discussed, the data you'll see today is using a research-grade Zeno-trap 7600 system in my laboratory at Monash University, Melbourne, Australia. And you've already given the talk outline, so I guess I've got the job of describing what the ZT scan is in terms of data-independent acquisition. I will predominantly focus on proteomics results and then finish up on some fairly preliminary immunopeptidomics results we have, comparing variable windows, Xenoswath, using quite an optimized method, to our relatively early attempts using the ZT-scan DIA, and hopefully I'll convince you that there are definitely some real benefits of this new scanning DIA method. So just to give you a brief technology overview, and comparing the two types of analysis. So the sort of swath analysis and the Xenoswath analysis, I'm assuming many of you will be familiar with, and this is a method where windows of ions are let into the instrument to essentially do a collective fragmentation experiment. So the data is collected in a step manner. The windows typically are around 10 to 20 AMU. They can be used in a variable method, so you can focus your analysis on busy parts of the mass range. And I guess another key element of this is that during this sort of accumulation of the window, that includes a TOF scan plus then MSMS of all the ions that are gated within this window, the collision cell needs to be emptied between moving to the next window. The ZT scan is quite different in that data is collected through a continuous Q1 scan. We'll go into the details of this on the next slide, and this adds the dimension of having the quadrupole, the Q1 resolution. And there's no need to empty the collision cells. This is really a continuous scanning process throughout the whole mass range. So the key advantage of this CT scan DIA really is this Q1 dimension. And what that does is it really, I guess, addresses one of the issues in data-independent acquisition, that is aligning the precursor mass seen in an initial TOF-MS scan to fragments that are then detected during the MSMS experiment, part of the DIA. So in the ZT scan, there's a stable transmission window of between 5 to 10 Daltons, and this is scanned continuously across the mass range. The initial experiment starts off with a TOF-MS measurement across that window, and this occurs multiple times over a chromatographic peak, which gives you your capacity to then do quantitation. Importantly, this MS signal is retention time aligned as well. Following the TOF-MS scan, there's this stable transmission window is ramped across the mass range, obtaining a quantitative Zeno-trap enhanced MSMS data set. And then we're able to align the MSMS data to that Q1 dimension again to get very good alignment of precursor ion mass and the fragments that belong to it. So how does this actually help? And this is work that's been performed in collaboration with SCIEX, in particular Ihor, and a PhD student in my group, Reza Dorvash. So what you can see on the on this side of the plot here, proteins identified from a 5 nanogram injection of K562 tryptic digest. We're using a microflow chromatography setup, fairly standard 5 microliters per minute, 15-minute gradient on a Phenomenex column. And we're using a Waters M class UHPLC. And what you can see is that when we compare the Zeno-trap DIA data, where we're obtaining on average around 2,600 proteins detected and quantified within this 15-minute run. When we then move towards the ZT scan, we see an improvement in the number of proteins we're able to confidently identify and quantitate. And importantly, we also see a 45% increase in the proteins that are quantified at less than 20% CV. So we're getting improved sensitivity and also improved precision of our quantitative measurements. If you look at it another way, well, what else can this sort of improvement in speed and precision do for you? This is comparing the same load of 50 nanograms of K562 digest, comparing ZT scan, which this time is on this side of the plot, with Zeno-trap acquisition. But the key thing here is that we're doing a 3-minute gradient on the ZT scan experiment compared to a 30-minute gradient on the Zeno-trap DIA experiment. And what you can see is that even with a tenfold reduction in gradient time separation, we're able to maintain essentially the same sort of quantitative information. So here we're able to improve our throughput without compromising the quantitative output of the experiment. The next thing we wanted to do to sort of really, I guess, test this system was to do a tryptic digest serial dilution experiment. So here we have various loads on column from 1 nanogram to 200 nanograms. We are working with gradients of 15, 5, and 3 minutes. So I think there's also some 1-minute gradient data on the subsequent slides. Again, this microflow setup. And we're comparing a few different methods here. So we wanted to be able to compare the ZT scan with the variable window Zeno-trap. So we're essentially using equivalent dwell times or accumulation times in the various windows. So we have this method here, for example, which is a 5 Dalton window, 750 AMU scan speed per second, which gives us this 7 millisecond dwell time. And we're able to have matching accumulation times in the variable window swath experiments. So these sorts of experiments generate a huge amount of data, and I wasn't sure which order to show the next 2 slides, but I've decided on this one. What you see here in these various sections are the different K562 loads, gradient time along the bottom here, so from 1 to 15 minutes. In the green is the ZT scan DIA data, and this is the proteins that we're able to identify in these experiments. And in the blue bar is the corresponding variable window swath experiment. And rather than sort of spend too much time drilling on this data, what you can see here is that we see the ZT scan DIA outperforming the variable window swath DIA, particularly at lower loads and faster gradient separations. You can see here, for example, with the high loads, the variable window swath method is actually performing a little better. Another way perhaps to look at this data is to look at it in a heat map. And here what you're seeing is the scale heat map here, and we're looking at the ratio of the corresponding ZT scan experiment to the variable windows experiment. So if the methods perform similarly, we expect to see a neutral color, such as here. If ZT scan is performing better, we'd anticipate that we would see green squares, and obviously if it's not working as well, we start to see these pink squares here. And I think what you can really see here perhaps more clearly is that particularly for these fast gradients and relatively low loads of material, we see superiority in both the total number of proteins identified and also the precision of the measurements we're making, with some I guess reverse of this trend at the very high digest loads on column. To further sort of benchmark these two methods, we did a mixed digest of human, yeast, and E. coli triptych digests in an LFQ benchmarking study. I'll just sort of briefly try and explain this experiment to you. It takes a little while to get your head around. So we made 7 pooled samples with different ratios of human to yeast to E. coli. We kept the human content constant at 50% of the total lysate biomass. To sort of, like, I guess, explain these ratios a little more clearly, for example, this 3:1 here would have 50% human, so 100 nanograms, let's say, in this case, and then a 3:1 ratio of the yeast to E. coli. So if this is 50%, then we need 200 nanograms of the corresponding digest. So here we have 150 nanograms of yeast to 50 nanograms of E. coli. This is explained in more detail in this panel here. I won't walk you through it, but as you can see, we're sort of generating these mixtures, so we can then go into the data using our 2 methods again, identical to the last lot of data I showed you. There are 2 gradients used here as well, and 2 loads, total loads of total material onto the column. The best way we could think of to really be able to, I guess, show this data, and I'm not going to bore you with all the data we've generated, just to give you a very quick tour of it, is this sort of distribution plot here. And so I'll just explain what you're seeing here. So on the x-axis here, we're seeing the intensity of a particular protein in the M1 sample. That is where there's an equal amount of material from all 3 of the digests. And as you can see, we have a similar distribution of intensities across the 3 different triptych digests we looked at. On the x-axis here, and these are all log2 transformed, by the way, is the intensity of the sample of interest relative to, I guess, that index sample. So shown here, for example, is the N7 data, where we have a 1 to 3. So we have 100 to 50 to 150 of the 3 different species triptych digests. And when we look at that, what we anticipate when we log transform the average intensity is that humans should be the same as M1, 100 in both. So we'd anticipate a log2 value of 0, and indeed we see that here quite nicely. Also shown here in a distribution plot. The yeast is around half of the amount in the reference sample, so we'd expect a log2 ratio, a log2 value of -1. And again, this lines up pretty nicely here with the orange-colored yeast dots. And E. coli, which has more. The log2 value should be around 0.6, which is precisely what we're seeing here. So we're able to sort of, I guess, get a good idea about the specificity and quantitation and the accuracy of what we can do in these types of experiments. So I guess to give you a very high level overview, just showing a little data here, what you can see is a couple of these experiments. These are all with 250 nanogram loads. And what I hope you can see in the blown up windows here is that when we compare the variable windows Xenoswath experiment to the ZT-scan experiment, what we see is the average intensities here are lying much closer to the dotted red line here, which is our anticipated value. So this is the 0.58 line here, which matches up very nicely with the ZT-scan. The Xenoswath data isn't terrible, to be honest, but here we're just getting that little bit more quantitative accuracy. And similarly, when we look at the Xenoscan data, the ZT-scan data for another ratio here, again, we see more consistent and accurate lining up of the median intensities here. So our conclusion from this experiment was that the ZT-scan has a higher quantitative accuracy compared to the variable window swath experiment. We've also looked at the performance of ZT-scan using nanoflow. We haven't got so much data for this at the moment. This is the setup we're using. Again, a 15-minute gradient, 150 nanoliters per minute. We're using a 9 optics SX Elite column, which is a 15 centimeter column, 75 micron. Again, comparing equivalent Xenoswath to ZT-scan DIA acquisition methods, you can see around a 10% increase in protein IDs going from these two methods, and also an increase in precursors that we're able to detect at different total loads of K562 tryptic digest loaded. So we're seeing that the same thing in the nanoflow experiments as we were in the microflow. Okay, just in the last 5 minutes or so, I just wanted to share with you the immunopeptidomics data as advertised in the abstract. So very quickly, for those of you that aren't familiar with immunopeptidomics, this is the study of naturally processed peptide antigens that are derived from typically in proteins within the cell, such as viral antigens, for example, tumor antigens, and so forth. These antigens are degraded into peptides, and these peptide antigens are able to bind MHC class 1 molecules and be transported to the cell surface. And we're interested in studying the nature of these peptides that are presented on the cell surface. The collective name for these peptides is the immunopeptidome, and it's very important in terms of controlling T-cell recognition of cells and with the potential to eradicate cancerous or infected cells, for example. The data I'm going to very quickly show you today is using a model of pancreatic ductal adenocarcinoma. This is obviously a devastating cancer, and this particular cell line was derived from a PDAC from a 56-year-old male, and has the HLA typing here, which is relevant when we start to look at the types of peptides we're able to see using the different techniques we look at. So the methodology we use to actually isolate these peptides, so we take cells, we lyse them in a very gentle detergent. This facilitates the solubilization of the class 1 complexes and in their native context. So this is a protein with its peptide antigen actually still bound to it. We then use a monoclonal antibody column to affinity purify the HLA molecules of interest. We acidify once we've washed this column, and this facilitates the the elution of the MHC peptide complex, and also the dissociation of the HLA molecules from the peptides. We use ultrafiltration to then collect the peptides in the flow through here and remove the proteinaceous material. And this material then goes on to our LCMS system. The experiments I'll show you today were analyzed with Diane. There are two sizes of injection we used, 2.5 million cells and 25 million cells. We used the same nano LC setup as you saw in the last experiment, same variable window swath and ZT scan acquisition methods. And we have a in-house generated PANC1 immunopeptidome library to search the data. So in a nutshell, and this experiment is literally hot off the press. What do we see when we look at these samples? So we see a modest improvement in the number of peptides we're able to detect here using either the variable window swath method or the ZT scan method. And I think that's probably a reflection of the fact that we need to do a little more optimization, particularly of the chromatography here. We see the anticipated length distribution irrespective of the method we use. And for the HLA molecules expressed by this particular tumor cell, we anticipate seeing predominantly 9-mers, which is what we see. If we look at the CV distribution of the peptides we're able to quantitate in these experiments, we also see a modest improvement in the CVs. So it may be a little hard to read here, but for example, this is CVs of 0 to 10%. And we see an improvement in this particular category relative to the corresponding variable window swath experiment. So we're seeing some improvement in the precision of quantitation using the ZT scan method. Reassuringly, the peptide ligand characteristics of the peptides identified with the different DIA modalities are very similar, as we would hope for both loads. So here we use a clustering algorithm to look at the different peptide sequences, which then correspond to ligands for the different HLA class 1 alleles. So for example, group 1 and group 4 here are two different subsets of HLA-A2 ligands. Group 2 represents the A11 ligands and so forth. So we're not biasing towards a certain type of peptide using the ZT scan relative to the variable window swath experiments. This is just an example of the sort of data we generate. And as you might anticipate, in both instances, we get spectra that match the library spectra very well. We also get quite good number of quantitation points across the various peaks here. This is actually a peptide from an HLA-A2 bound peptide from the PRAME cancer testis antigen. So we're also detecting peptides that are potentially actionable in the context of immunotherapy in this particular cancer. So just to sort of quickly summarize, improved speed and precision of proteome analysis using ZT scan DIAMS. I think we've shown that pretty definitively. In terms of the immunopeptidomic analysis on this system, there's a definite need for sensitive qualitative and quantitative assays to detect cancer epitopes, and we're certainly able to detect a number of cancer epitopes within our data set. We've actually developed the variable window Zeno-trap DIA as part of a clinical pipeline to map tumor-associated antigens in clinical specimens and biopsies, a paper that's just very recently been submitted. But what we're starting to see is that CT scan DIA is showing further improvements in precision and sensitivity for these immunopeptidomics measurements, and we anticipate we'll be switching across this modality for our clinical studies in the very near future. Just remains to thank the people that were involved in the study. I've sort of mentioned most people as I've gone along, colleagues at Ion Optics, a number of colleagues within SCIEX have been very helpful with early access to the technology, both in Toronto and also in our local guys as well. Reza Dorvash has done all of the experimental work in my laboratory under the supervision of a fantastic postdoc, Patricia Illing. Thank you for your attention.